Meeting and great. So this is another meeting of Agential topics. I'm Charlie Munford. This is put on by Talking Octopus, which is our organization that's dedicated to building a physical model of agential biology. And today we have a short presentation from Heng on his theory of karyotype codes and two-phase evolution that we're all very excited about. So welcome to everyone, and anyone who's new, maybe say a word or two about yourself just to introduce it into the recording, and we'll go from there. Let's see. I guess we've got Dennis, Marco, and Rafe are all new. If you guys just want to just say like 3 sentences about who you are and what interests you about this topic.
Sure. Hi, everyone. I'm Dennis. I'm immunologist, stem cell biologist by training. Currently run a synthetic biology company called Prolific Machines. Very interested in all things karyotype.
Awesome.
Okay. Thank you, Charlie, and everyone for inviting me to this fascinating discussion. And I'm Marco Cavaglia, MD anesthesiologist. I'm currently affiliated with the Polytechnic of Torino, and I'm deeply engaged in interdisciplinary research spanning from neuroscience, molecular biology, and biophysics. The core of my work is the lipid-centered theory of brain functioning, which reinterprets the role of cellular membrane, particularly lipid bilayer, not just as a passive barriers, but as active players in information processing.
Awesome.
We were just talking about this. I was just saying we need the guy who does this with things other than, well, anyway, who looks at it with membranes.
Totally.
Thank you.
Good to meet you.
Yeah.
Nice to meet you, Green.
Oh, sorry, I'm going to change it. It's Marilyn.
Marco and
okay, Marilyn.
Marco and Perry Marshall are friends too.
Oh, great.
Yeah.
Hi, everybody. I'm Rafe. I have a long-standing interest in complex adaptive systems, evolutionary theory. I met Henry about 20 years ago, and he blew my mind about genomics and cancer and agential topics. And I've been a fan and advocate ever since. And a couple of years ago, I published a piece highlighting what I feel are some major breakthroughs in from Henry's work. And I'm good friends with Mary Lynn, new friends with Dennis.
Rafe thinks we're good friends.
Well, burst my bubble.
I humor him.
Yeah. Anyway, I'm really excited to be here. Yeah.
Fantastic. Terrific. Henry, take it away.
Who? Steve Lahar.
Oh, okay.
I'm interested in harmonic resonances and this idea of the DNA being unwrapped in segments to vibrate freely by Brownian motion that creates electromagnetic waves that reach out over time that are very, very interesting to me.
Thanks, Steve. Fantastic. Terrific. Henry, you want to take it away or?
Okay. So so can you see the slides now?
Yeah.
Yes.
Great.
Okay. So first, thank you so much for participation. And I actually just besides Reef and everyone else that we haven't met before, but some of them, like Bill, we actually have communication, but never face to face. Hi, Bill. Yeah. So I'm very glad to briefly tell some of our ideas and in the purpose of get some good feedback and advice from you. So today I'm majorly focused on karyotype, but to thinking about this is a new way of the information platform, and really go through the two phases of evolution. So the idea is how the evolution basically lead to increase the complexity of information, both the information themselves and its carriers. So that's the emphasis for the current study. But before I get into this detail, I would like to introduce the system I'm using, because currently most people are not familiar with the cytogenetics due to the domination of molecular biology, right? So for the status of current biological research, we know the big data is coming, and we know the limitation of gene-centric idea, and we know we have so many beautiful theory, but it's hard to apply in the real world. So the new approach, people emphasize technology, sequence more, and doing single cell analysis, spatial temporal analysis. All of this is good, but there is a key limitation is because we really need to have some solid biological based theories, because we have so many assumptions, but not really experimental testable ideas. So that's what we have been done in the many decades now, 3 decades or more. So the problem I would like to focus on is really some of idea we never truly tested them, right? So for example, in genetics, we have favored the gene-centric idea for a long time, right? But now we know there is a limitation because the inheritance we cannot be found, right? People simply jump into the epigenetic, but it's not that simple. So we try to find the button of the truth. For the evolution, even most important mechanism like the microevolution accumulation become macroevolution, there is no evidence, actually solid evidence to show that. And but people just truly believe it. So that's why we actually really need to raise the question. And more importantly, we realize that there are so many different specific mechanisms which are historic context dependent. So people just simply use one of the mechanism to push the universal evolutionary theory like Darwin did. And many people try to use an alternative way to reshape the evolutionary theory that all goes to the same way. But as I will point out, because they have so many different specific mechanisms, so we need a new direction of how to unify them. So therefore, for the future biological research, for my part, I think most important is really have some solid concept, and from where we can build the different ideas. So so what I try to say, let me change a little bit about this one. Are you still consider slides?
Yes, absolutely.
Okay. So this is better for me, because previously when I put the full screen, so I only can see half of my slides. So I don't know what I'm talking about.
That's fine. We can read it fine, just like this.
Okay. So now it's good.
Be able to see you too, Henry.
Okay, that's good. So for the cancer evolution, the unique part is a very powerful platform which can actually test the previously some of our idea. So for example, we can directly examine the relationship between phenotype and the genomic type, and the key is the process, right? So for the phenotype, we can know how the cell growing, cell death, the advantage of growth. But for the genome type, we can study different scale. We can compare, gene mutation, compare chromosomal change, and compare the transcriptome. So all of the phenotype. And the key is really doing the experiment which have different historical event and we can repeat multiple times. We can do in a parallel experiment. We can see the transition from individual cell to population to really test how the evolution actually occur, how the genetics is driving this process, right? So I will briefly just tell 5 stories, each story 1 or 2 minutes, and to introduce the system. So the first story is we trace the cancer evolution process and discovered the two-phased evolution. So the system is rather simple. We have the culture cell from normal cell become cell with the feature of the cancer. I mean, this whole process costs 2 to 3 years. So we continually culture those, and we continually every 2 weeks check the proportion of the population to say what's the involvement of the gene, involvement of the genome, and the involvement of the phenotype. So from this different stage, initially we try to validate the Darwin's mechanism of the evolution, but as I'm going to show you, the results just blow us away because many of the assumptions beautiful but not true, right? So this is just the one of the figure in the paper you have read briefly, but I just try to walking through with you. So this is a chromosome painting by different color. So we use a sky technology to paint each chromosome with one color. So therefore, any chromosomal number change and translocation, we can quickly score them, right? So at the very beginning, the top panel, you can see this is the early culture. So they already have some chromosomal change. But if you follow this progress that's in the middle panel, and all the new chromosomal change is not directly inherited from the previous one. So we know the cell this middle panel is the results of this early panel. The DNA is similar, but the chromosome is very different. So that gives us some initially idea. We think, okay, the DNA clone and the karyotype clone is different. So what does this mean at that time is very puzzling. But it goes through this whole process from pre-immortalization, become immortalization, become post-immortalization. Major change every time is at the chromosome level, right? So that there is the same DNA to build a different structure, right? So that's the idea surprised us. So we put the 3 years of the data in the one slide, so you can show you they have different color of the pie. So different color showing is a clonal chromosomal change, right? So they all discontinue, right? And then we can see the peak here is a random chromosome change. So you can have more and less as such. So if you're talking about the phenotype, we know the PD7 passage doubling time, right? Different stage. So you can go through this process. And then we can see the random chromosomal change just become peak and go to this way. And the karyotype themselves is discontinually jumping, from green color to purple, to orange, to blue, and to red, right? So mapping back, we know here is a crisis stage, is most of the cell is dead, right? But then some cell be selected upon, and when they are stable, they form the clonal population. So like the red pie you can see here. So the chromosome no longer change in about 2 years. Only gene is changing. So from this simple analysis, we actually suddenly realized initial evolution is jumping by the chromosome, but after the selection, they become same chromosome, but a gene mutation epigenetic change. In other words, initially major change is chromosome discontinue, but after that is a gene mutation continuous accumulation, right? So what we have found is the red color pie here. This is a Darwinian accumulation type of evolution. And before that is not driven by gene accumulation, by small change accumulation become big. is totally different phase of the evolution. So with this phenotype correlation with this, we know the transition actually from random to the pattern, this phase transition is completed by transition from macroevolution to microevolution, right? So that's the whole story. I hope you get the idea is there is not a simply Darwinian evolution, but Darwinian evolution just a small proportion of this whole story. So to make this very simple in terms of understanding, we say it's different shape, that's a different genome change. And then same shape is the same genome, but with just different gene mutation accumulation. So now we divided the evolution as a two phase. One is a punctuated phase, one is a stepwise phase. And the punctuated phase corresponding to macroevolution and corresponding to creation of the new system. And the stepwise evolution or Darwinian evolution just corresponding to microevolution and corresponding to the growth of the population, right? So that's now we understand evolution is two phase. One is individual, one is the population, right? Rather than the previously thinking the population replacement, which never happened, right? That's another thing. If we study the fossil, we'll get to the conclusion. So now the conclusion of the first part is simple: from normal cell to transformed cancer cell, from non-invasive to the metastatic, from drug sensitive to drug resistant. All of this have to go through the new karyotype formation, the process. And then the gene mutation as the different color of the arrow here, stochastic associated with this, just help the population growth, right? This is very simple about the cancer story. So the second story is the genome chaos, because we actually showing the cancer evolutionary formation that have so much change. One of them is the genome level drastic change we call the genome chaos. And what's the nature of this? We spent almost a decade to study the phenomena. Now we realized this is a mechanism to drive the phase transition from the phase of the create new system to the phase of just simply duplicate this system by growth, right? So actually the people outside the science field understand the chaos very well. They're thinking that all the great masterpiece is just by disorder of the dictionary, right? You just created this process. So this is much more understandable than scientists. So what looks like? So you can see the three panel of the chromosome. One is a cancer cell before the drug treatment. So each chromosome have their own color, but after treatment in 24 hours, they become fragments that rejoin, so they totally form the new monster of the chromosome. And the big panel is a number change. Sometimes one cancer cell could have their 1,000 chromosomes that together they continually change. And now we understand this is just the process of create new information and sort the information out. And if people read the sequence paper, you can see this pi that connected by different chromosome. That's this crazy chromosome. If you sequence them, they're just like that. So now, if you read all the cancer paper, most of the cancer just like that, just genome chaos. So in fact, in the past 30 years, we have studied each different this type of crazy, chromosome change. People always thinking this is low use information, but is fundamentally important in the patient sample. This kind of structures everywhere. So the cell can become giant cell. They have the one cell dividing, one cell become over 100 cell, completely dominate the process. And sometimes the whole genome can form the entire genome. They can form the one of the chromosome over there. So anything you cannot imagine, it can be happen, right? So that's why the cancer evolution is so difficult to fight. So here is just one of the process, one run of the about 6 months of experiment. So the panel A is the cancer cell before treatment. Panel B is the treatment after 24 hours. They just become chaotic. and they quickly form the panel C, they form the totally different chromosome. You can see that each chromosome from different number, 15, 13, 5, 7, 8, 9, 14, they just form the new chromosome. And they continually unstable sort, and finally become drug-resistant cell. If you analyze them, they form the clonal, however, is much simpler. So the dramatically chaotic the intermediate structure is necessary, but most of the time is not visible after the formed, the drug-resistant progress. So we actually imagined, proposed the order of the gene A, B, C, D, E. They have a certain order, but after the genome chaos, they change the order on chromosome and they change the new information, they change the karyotype coding. So that is what's going on here. But of course, for over 10 years, people always deny this. They say it cannot happen because they thought the biology is so precise. If it's so disorder, how could the biology can be possible, right? But that is really happening. Almost 10 years ago, when people sequence all the crazy cancer, they found it's a universal phenomena. They said, aha, it's true, but always give different name. So right, Nature Cell Science have 34 names now. They refuse to use the genome chaos. They just use their own terminology to describe the same thing. Like the Harvard MIT group, they just use a genome, use different dark night. We just use ABCD, and they just the invent the well of all of this study. But that is what happening right now for the genome chaos. It's become very important subject. So we actually realized all the different the polyploidy and the chromosome fragmentation, all the crazy stuff just in the step 2, just to create a new configuration of a chromosome. And then the population growing just by the duplicate the already survived with advantage. So all the cancer process is just like the evolution process, is an evolutionary process, is the information process, right? So because of that, we actually have to ask the question, why they want to arrange the chromosome, right? So we actually realized because there is a new coding called the karyotype coding. Because we're showing that we try to understand this process. Every time that we reshuffling the chromosome, they actually change the order of the gene on the chromosome, right? So that's the conclusion we got. So therefore, we're thinking to understand the powerful change, we have to be able to understand the underneath of the evolutionary process, which is create and select the right information, which is allow the information carrier can grow in to carry on such information, right? So with this kind of study, we really need to understand the new genomic coding. So we know the gene coding because the ATGC coding, the from DNA to RNA to protein. We know that, but we also know that that gene coding is not at the system level, is not the blueprint, right? So then we actually divided the information at the 2 different scales. One is the gene, is the parts information, and one at the genome is the system level of information. So we use a metaphor to say almost like the brick. You can use the same brick, and you can have different architecture, build a different house, pave the road, doing a different thing. But we also know the configuration of the gene in the nucleus is not random. They have the certain configuration. We think in such a configuration is the structure basis of genetic network. So that's our idea. So if you same gene but different configuration, you have different structure basis for gene interaction. Therefore, you have different gene expression, right? But this appears too simple. Even the Eric Davidson, all this pioneer in system biology, they think this just is too simple. And we're thinking there is no such a structure basis, right? But the more and more people realize, yes, there are, because the biological system is different from pure chemical system, and they do have the configuration, they do have the impact of the function. So we just give some example. Just so many examples. We just use a sponge. Sponge have 18,000 gene, right? they have immunogene, they have neurogene, but they don't have any tissue differentiation. So we thought that actually just in the wrong configuration. So if we propose the experiment, if people can, use the genome chaos in the sponge, maybe sponge could immediately have so many advanced feature. So that we're really showing the importance. And of course, we know if you just introduce one piece of chromosome 21 into the cell by microinjection, and another part of the genome expression also changed. This is not just a simply dosage effect in the certain region, but the whole genome is changing. So the logical explanation is yes, they do have the, the coding, and also Hox gene cluster, everything you change the configuration, and the embryogenic become lethal. So therefore, evolution is select this configuration as well. So now we know, if people actually sequence all the animal from initial to the, also the, flower planning, every species is just by change the configuration of a chromosome rather than by invent a new gene, period. But people still resist this idea because, anyway. So we actually also realized, for example, cancer research, we realized the karyotype coding is the key for macroevolution and spatial aging, because now we are not talking about cancer anymore. We just learn from cancer, and so let's look for the, the fossil record from the, the species formation. And the conclusion is the same as the cancer. For example, traditionally people thinking species A like a square, that gradually change and becomes species B like a, like the circle, right? So gradually this process. So the challenge is they cannot find the intermediate structure. So people just say, oh, fossil is not sufficient. But now actually we actually realize initially change like the species population is by 2 individuals, right? So just one generation, they already from square to become circle, but they need a long time to duplicate the circle, rather than previously thinking need a long time to gradually change, because we know the change is all of a sudden one generation finished. But then most of the time, they don't have a chance to duplicate the population. So that's a new model. And with this new model, if you see the evolutionary data, it's completely this case, right? So from the big birds, from all these plants, and just exactly like the cancer story. So now is the time to change the evolutionary theory, but people still, resistant to do so. So recently, we actually, recent few years, we actually focus on the information innovation and the conservation. Surprise, we found that most of the time, evolution is conservation, is not innovative, right? So innovative is by macroevolution, and the vast majority of the proportion of the time, what evolution did is to preserve the information. Do not let the information change. And therefore, you have a chance to duplicate the same information. So therefore, what we have, clear picture is really thinking about how the behavior of the information and also behavior of the carriers. And surprisingly enough, you need a crisis, the environment crisis put the pressure, and then therefore genome can change. They have the opportunity to change, and plus macroevolution. As soon as this package be selected upon by macroevolution, the immediate switch to the microevolution phase is evolution is all about preservation information and use a biological process to duplicate the miracle, right? So that's what all the evolution about. So that's a big picture of evolutionary theory. So now we actually realized the information management is important because they have the 4 components. The first, you have information self-creation. So you have to anticipate it by the biological process. But if the biological process is very, very early, they have limited the capability. And gradually they have more capability. And the cognition become more powerful, more information rich, and finally reach to the consciousness stage. So all of this is information increase the complexity, right? So if that way we understand this process, then we actually really understand the importance of the preservation of the information. So therefore, biological system is the system can very nicely preserve the information by inventing the inheritance as a system. And the karyotype inheritance is one such example because they can host so many different codes, right? As a carrier to get 1 layer, 2 layer, 3 layer of complexity, and completely use this complexity to ensure the information flow, right? So that's all about. So after that, they have the information modification and they have information usage. Those two things is mostly involved gene epigenetic study and the most involved current information study. But now we really emphasize the self-creation and the self-preservation is fundamentally important, if not more important than self-organization. Because after self-organization, you have to be able to preserve such a process. Otherwise, they will go back to the nature. The complexity will never be increased, right? So that is the key for us to thinking about this. And during this process, many biological process, the function we have to rethinking. Just give you one example for the function of sexual reproduction. Previously people always thinking, parents are different, they produce diversity. But now we realize actually is not the main function. The main function is the preservation of the information because the order of the chromosome during the chromosome pairing in meiosis, they just checking the order. If the order is wrong, they just automatically abort it. The system is off. So therefore, meiosis, the key is make sure the species never changed in the normal situation. So therefore, they can completely duplicate, but still same species. So when we initially proposed this idea, the people in the genetics field is just shocked, right? So this is completely impossible after 130 years. And then later on, the Ruby Holiday and Adam Waking, they rethinking this, the idea, they actually get the conclusion. Surprised, they actually realize that the initial function of chromosome pairing, it is reduce the diversity rather than increase the diversity. So now, I mean, all of this idea need a complete change and to entertain this idea. So we actually did many of the experiments showing all the dynamics, how the information, they come from the, small cell, the fusion from big cell, and then resorting the chromosome. This whole process is called the, polyploidy, the giant cell, all the information. So on the video, you can see the cell dividing, just one dividing to become large number of cells. And each of different cells, they have different chromosome composition, and the evolution just select the winner. We actually calculate the rate is about 1 in 10 million to 1 billion rate. Every time they have to have the new stuff, they will form, and they will become dominant. It's a process. So lastly, then we actually really thinking for the evolutionary theory, we really need the information and the historic contingent model of evolution, because each different stage of evolution on the Earth, they use a totally different mechanism, right? So the Darwin, actually, he majorly used his argument is artificial selection. Artificial selection is much late in the evolution process, right? It's very, very different. So when he used artificial evolutionary conclusion mechanism to explain the whole entire evolution process, he made a fundamental mistake, which we can discuss that as a lot. So nowadays, people actually manipulate the information, like they are doing the symbiotic study, to inject. I mean, Riff just sent me a few days ago the paper talking about how to use inject the bacteria into the fungi to see the relationship. All of those is use artificial information mechanism. which is fundamentally different from how the nature is operating. So that's another story. But anyway, so now from the different biological physics and the chemistry and the different stage of the biological evolution and the social evolution, cultural evolution, they all involve different codes, different law. So that's the key point I want to say. And also the new frontier, we think most important is to consider cognition and consciousness is a higher form of information carrier and the information form. In that case, we can actually integrate to culture and technology evolution as the biological evolution. And also because of that, we actually very worried about the current AI technology could become the carrier to replace the human, because it's not about who is smart, it's about whether or not we have a competitor for the carrier for the information, right? So that's totally different from the thinking, which we have some new model as such. And I will stop here and have some acknowledgement. So during this time, and so many people involved, and help me to maturate our idea. So I will stop here. I hope I have give you some idea, but I really want to, get some feedback and advice from you. And so sorry for a little bit longer, but there's too many different aspects. Okay, thanks. I will stop here.
Henry, I'm so glad that you quoted Cocteau, since I always say that the true scientists I've ever known have the soul of a poet, or at least an experimental surrealist filmmaker. I really appreciate that.
Okay, thanks. Yeah.
Fantastic. Henry, I love this theory. Thank you so much. This is really wonderful. And I've enjoyed just appreciating every part of it. I have a question for you that's related to the times of crisis when genome reorganization happens. Because I'm trying to resolve in my mind, I'm trying to paint a picture of like the conditions of stress that cause karyotype reorganization. I'm having a problem because I can imagine time, like the word stress is ambiguous for me. And I can imagine forms of like hormesis that I expect actually reduce cancer. So like fasting or exercise, are like forms of stress that clearly like create pressure for cells, to complete their metabolism in various ways. And yet they seem to reduce cancer. And so I'm trying to unpick in my brain the way that like what constitutes a crisis for a cell that would cause this reorganization, and how does that sort of play out?
So actually, this is a good question, because on the surface is very paradoxical, right? You're thinking, but I mean, just before I give you explanation, we can consider immuno, system as an example. So immune system actually will prevent or reduce the cancer, right? But fundamentally, if without the immune system, we may not have cancer in the first place. So that's actually a story of biological system. Everything is interact with each other. So if you don't have the stress involved, then you don't have life, right? For example, if you lock out the AR stress pathway, they just cannot finish cell differentiation. So we need the stress to make life, but when we deal with the stress situation, the life to pay the price, we got disease. So we actually cannot simply say who is the cause, who is results as such, because in such a complex system, the causation maybe is less important as we have been told to think about that. So for example, in this case, for the cancer, for example, so if we try to treat the cancer cell, right? If you put the stress, the always will reduce the cancer's growth, right? So that is true. But then we actually try to kill all of the cell. Then we trigger, when we use the maximum dosage, then we trigger the cell, go to the crisis stage. And then they become different game now. So you cannot continue thinking about previous game. So the complexity emerged to a new game. This new game is whether or not the cancer growing well. They're not growing well. They actually reduce to the maximum, right? But because of that, the new game, therefore, the new game is none of the cell, including normal cell, can survive. And then at that time, the cancer, the monster, that becomes so powerful. Right. So for the cancer research, people actually miss the point. Most of the cancer, if you do not treat, many of them is automatically dying by themselves. So people fooled by this, they say have a new drug. Oh, half of them killed by my drug. No, if you don't do anything, half of them were dead anyway, because that's the dynamics of how the cancer win the war. They just sacrifice majority because such a sacrifice have loyalty, because that they can win. Otherwise, by average game, cancer is never going to win, right? Because you have billions of normal cells, they will kill them. So that's a totally different way of thinking biology, because so I did not directly answer your question, but now I need to directly answer your question. Directly answer your question is yes. So in the normal cell, like the liver cell, people did experiment. The cell have a beautiful 46 chromosome. After baby start drinking, the toxin is there. The cell cannot finish that. They become polyploid. They become aneuploid, because that's a way to adaptation. But if you have too much adaptation, right, you great, everything, you solve the problem, but what's the price to pay? The price to pay is the configuration have different configuration. And the different is the key for evolution, right? If they're identical, they don't have evolution, right? So with the different, so therefore the diseases, most of the time, the byproducts of the cellular evolution, right? So that's a basis, but then someone is lucky than the others as such that were different. So for example, when they have p53 mutation, you have the family, you get the cancer is much more frequently, right? You have Liphium syndrome, everything. That's very bad. But imagine there's a nuclear war and only this family will survive and the normal people will be wiped out. So that's the game of the life. I mean, you don't know which one is good, which one is bad. Everything is contact dependent, right? So you're talking about autism, and it's a problem. They're not social. But 10,000 years ago, everyone is working in the field. You do not need cooperation. You do not need to have articulation of everything. So you don't have the phenotype of the artistic or something, right? So all of this is historically contact dependent. So I cannot give you defined answer, but I only just surrounded this question to tell. But we know for the speciation, most of the species formed is a during massive extinction time. That's a fact. And we also know this for cancer therapy right now. If you gradually just kill small proportion, the karyotype will not change. But as soon as you want to eliminate them, use high dosage of maximum killing, you trigger the genome chaos. So that's why we actually try to use this idea in the future, to use a very moderate treatment to control the population of the behavior without trigger the survival of genome chaos. Yeah, that's another thing. Yes, Mary.
So your theory just opens up so many things for me and explains so many things and is so homologous to other processes that I see happening in populations of all at every level of the holons of life. For example, when extraordinary population pressure is put on a population of wolves, for example, they change their biologically, they change their reproductive behavior, and they produce more offspring more quickly. And it seems to me like you're explaining a lot of things that you're echoing a lot of things that I see we could probably apply at different levels. But one question that I have is, I'm when you're talking about, you showed the slide that showed the newly reformulated chromosomes, and in the first saltatory step of evolution, and then those having to somehow reemerge in other cells in sexual selection in the opposite sex in order for them to be able to actually combine and to create a new species. And I'm wondering if there's ever sort of a mixing of polyploidy and the creation of the new chromosomes, so that maybe a whole chromosome is sort of a partial parthenogenesis, so that a whole new chromosome is transmitted to the opposite sexual partner, if that kind of thing ever happens, if I'm making any sense.
So they have some similar, make sense, in the plants. So the plants, actually, people hybridize with different species, right?
Yeah.
So they artificially put them together, they often form the polyploidy.
Right.
And after that, the polyploidy is unstable. They actually emerge many, many different strains.
Yeah.
Only some can be survivable when the chromosome is stable. So
Right.
if they're not stable, they actually can temporarily survive, but then one or few generations are dying off. So those become invisible for us. We only can see the survived stuff come out. So there is a story and a similar story. And plants, we know, is everywhere like that. So the similar story is a human. Human, we actually still have the chromosome 13, 14, 15 fusion, because the acrocentral chromosome can fusion together. So the frequency, the baby is one in a few thousand baby, you have this one. But because these two chromosome fusion, they don't have much phenotypical. We anticipate if in a different environment, they will have a huge difference.
Yeah.
So, but, the come and go, people will still, someone have phenotype, but the most, if you have a phenotype, is always involved, the intellectually is little bit, off, but still okay. But in some family, they involve relative marriage, right? So we check the literature. So in the 70s to now, 60 years, they have 3 cases, one in China, one in Middle East, one in Europe. So they have relative marriage. So the chromosome, the 45 chromosome become homozygous of 44 chromosome, right? So by our definition, they already become new species. But the good news is they don't have a partner with the same 44 chromosome. So they get the 46 chromosome, so all the baby are automatically aborted. Yeah. So this 3 story all finished over there. But imagine if these people in the island and they actually absolutely will form the new form of the species. And then if the climate change so dramatic, maybe they become only can survive and we wipe us out. So that is completely is possible, especially right now to thinking about this story. Yeah.
Have you looked at Luc Montagnier's paper about the transmission of genetic information using harmonic resonance and structured water? Because we should have a conversation about this another time, because there's this wild universe of thought in which a whole chromosome could be transmitted via harmonic resonance from one cell to another, and not have to be actually, so maybe even a chromosome changed to be homologous to one in the partner gamete. We should talk. It's an interesting idea. With something that our little research group is looking at. Marco's shaking his head. Do you have something to say? I'm feeling like I just put out the dumb idea, and I want somebody else to say something that makes me seem less insane.
Thank you, Marilyn, and thank you, Dr. Heng, for your sharing your beautiful theory. And according to water, I'm actually really interested in what Dr. Heng was calling the new information carriers. And
Yes.
thinking about water, if we take a moment to consider something that's seemingly simple but profoundly impactful, the role of water in biological signaling is a topic unbelievably important. And is often overlooked, water, and is not just a passive medium, but an active participant in critical biological processes. And as we know in biology, everything's happened in a liquid phase transition. So water as a mediator, for example, of electromagnetic field, due to the dipolar nature of water molecules per se that allow to align and respond to electromagnetic field that are generated, maybe even by chromosomal structure. And chromosomal structure, just according to the phosphate group, just delivering all the negative charges on the backbone, create electric field that are already measured. And these aligned water molecules can create a coherent network, enhancing the propagation of bioelectric signal across and within DNA chromatin. So actually, we are talking about a structure that is going from molecular to functional. So it's crossing dimensional space, but it's still the same structure. So there is to be different level of information passing through, and maybe even a transverse way of information interconnecting the macro to the micro. So actually, water and the dipolar property of water could really be an interesting medium that allow for a better explanation in understanding what is really happening from the molecular realm going through higher dimension, especially when we deal with information. So the water, especially related to the DNA, is not only creating the hydration shell around the DNA, and actually it seems that the DNA has the shape that has only when it is hydrated. But moreover, water and DNA is very harmful, since if I'm correct, the backbone need to get rid the bases from water, because actually hydrolysis is one of the first mutation process for DNA. So it's something extremely important that maybe we don't have yet the right instrument even to measure and to look at. But according to even indirect evidences like the Montagnier and Preparata and Vitiello, I mean, there are many evidences in basic research science about the possible role of structural ligand water in processing biological information.
So now I actually understood. So that could be one of the general mechanisms. For example, how the information. So which is slightly different from what I'm talking about. Is at that level, they actually have the coding the information specific for the species specific. So I guess different species, they could use the same water DNA interaction mechanism to deliver that mechanism. But here, what I'm talking about, I did not address whether or not the water interaction will have the species specific manner, but rather than, so this coding we're talking about this is really focused on the different species. they could use similar biophysical, like a water interaction with that. That's is absolutely, yeah, it's, but it's from different angle as such, yeah.
I want to just ask a question that synthesizes what Marco just said with Henry's idea. It seems to me, if I can translate this to high school biology, so forgive my simplicity of mind, the Luc Montagnier, Pollock, Mei-Wan Ho, and the Italians who've been looking at this are putting together this idea that the water acts like a sort of negative template for the DNA or the strands of the DNA or the whole chromosome that in some way acts like a resonator or a antenna. And then that structure that's outlined by the structured water is then transmitted quantumly, or at least over short distances, either atemporally and aspatially through our quantum methods, or at least over short distances to other structured water. And you can have, if you have nucleic acids lying around, you can have a restructuring from one cell to another, one gamete to another, and possibly have the reorganized chromosome transmitted in a non-direct physical transfer of material type fashion, but rather an energetic fashion.
Okay.
So this is the idea that Montagnier comes up with, and that's what I was leading to. I was saying, is there ever an opportunity where the saltatory evolution happens really fast because the newly constructed chromosome that has come about through gene chaos is able to be transferred quickly to, I mean, we know that that individual embryos, even, you know, just when they're only at the 2-cell stage, can communicate with one another in a multi-embryo, you know, setting. Why couldn't there be this level of communication between the gametes before they even come together to create?
Sure.
Yeah.
Sure. So I just for the sake of argument, right? So I would offer, you know, slightly different explanation, because now I think many people put too much effort for the quantum physics to explain biology, because
Okay.
from our view, we actually found that each different scale, they have their own law as such.
Okay.
Because they have alternative explain to this phenomena, because we know that cell differentiation, even the same genome, they have different, differentiation marker is clearly there. And for example, the chromosomal configuration, genome chaos, every time when they become evolutionarily successful, they just copy the cell. So the biological use biological process, not the physical process, to have beautifully finish the job. So that's why I emphasize, the biological is a system which can turn to the miracle into biological routine, because they just use cell dividing. Every time they have all this stuff, right? If initially have this chaos, if stable, every cell has the chaos, right? So they do not need the complicated, physical to transfer some medium to pass information. They have direct information already in that cell every time. So that is one of the important part. For example, people talking about the tangling of the quantum and to explain neural phenomena. Actually, the way much to emphasize this unsolvable, but in fact, they have biological basis, solid biological basis actually can offer if we are in the right, the angle to talking about that.
I agree with you with that, Henry. I think that there's a lot of jumping to a metaphysical and mystical solution when we have a direct processes that can explain things. However, seems to me that life is mysterious and multidimensional, and also there's a lot of redundancy in systems and echoing of systems, like energetically and physically. So I wonder if there can be multiple ways that genetic material is transmitted.
There could be, but on the other hand, right now I'm more
But let's
emphasize.
not throw out the baby with the bathwater. Let's look at all of the mechanical structures we have in front of us and exhaust our understanding of mechanics and physics as we know it before we
Sure,
jump to all of
sure,
the metaphysical,
sure. But
aka quantum explanations.
sure. So for example, I mean, you explain if there is no biological basis, I will go for that. I mean, it makes sense. Makes
Yes,
sense.
that's right.
But now when we have a multiple causation complex,
Yes,
what I have learned is we have to prioritize.
yes,
So for
point.
example, if we now know the life and the non-life, what's the difference is the information management capability.
right.
So quantum, for example, the Chicago group that did the simple physical phase transition, they think, oh, behavior looks like a neural network. Looks like, but it's different, right? So therefore, when we read out, we always see something different. Then we use one mechanism to explain that actually can have many misleading information because during for example, during the whole process, we analyze each different layer of the prioritize. They're all different. After the genome cannot change. The epigenetic become very important. But before that, there is very minor, but it's still involved.
Right.
So that's why. So when they involve
good point. You
physics,
can't be looking at epigenetics when what you're looking at is a whole reorganization and creation of totally new morphology of the chromosomes and number of them and so forth. Why look at epigenetics at that point?
Yes, but after social behavior, whatever, right?
Yes.
So the biological genome become less important. So what we are talking about here is just one type of window opportunity. So now we actually divided the history into 8 or 9 totally different mechanism. For example, the fusion of the cell from mitochondrial capture. Most important is at that stage. Later on, the happen is no big deal because the biological routine becomes so dominant. We do not need to invent a new well as such. So that's why the name emphasizes that to trace that on. She also made a mistake about that. So now we are very clear is about the genome chaos, which most dominant is in the complex biological system. But when from outside the biological domain, it's slightly different now. This another force is in charge.
I want to ask you a question about the moment when the gene chaos is happening. Have you heard, or has anyone else looked at the properties of the condensate or the coacervate that's caused when that's happening in terms of the interaction of maybe structured water with, I mean, this was Mei-Wan Ho's theory. She didn't really flesh it out very much. She just put it out there and she said future researchers will have to explain this, of how there's some kind of structuring or directing of the reorganization. She was just talking about it in mitosis and meiosis in general, but is there something about the unique way that the cell is directing the reorganization of the chromosomes in that condensate or coacervate phase? And I'm just wondering if I'm just working on a very general hunch.
Sure. So we actually realized that for so many factors in biology, right?
Yeah.
So they actually can become same language is a stress response. That is true. So for example, if they change homeostasis, they change everything. So at a certain stage, the cell become unstable. The unstable, they will trigger chain reaction of even more unstable, right? So that is so it's quite possible because we actually link to we've spent years to link to the random chromosome change. What's the random chromosome change? Now we know everything can cause random chromosome change, right? So you have nutrition, you can cause radiation can change, drug treatment can change, even psychological can change.
So there's a positive feedback loop of destructurization that goes across materials.
Anything, if cell sense the variable, they will actually become the stress.
Yeah.
Right, as such. So that's actually universal unified theory. We always thinking is a stress response as such. But free response have two outcomes. One is good outcome, one is bad outcome. It's totally unpredictable. The always, if it's not good, if it's bad, right? Or doesn't matter. So therefore, the genome unstable is one of the key link with whole system as such.
Richard's had his hand up for quite a bit.
Oh, yes.
And so is Willie, and so is Bill. I'll shut up.
Richard goes first.
You need to turn on your speaker.
You're still muted.
I waited too long. I forgot. Thank you, Henry. That's been really fascinating, and I'm a little bit overwhelmed by all the new information because it's still sort of percolating around. But building on some of the things that you were just discussing, I'm curious about your view about the randomness of the chaotic stage. So the sort of the neo-Darwinian model of change is that change happens at random and then selection keeps the things which work well. And one could interpret some of your observations as a Darwinian model of that kind, but at two levels of organization, right? So that there's a random karyotype reorganization thing going on, and selection works on that. And there's also a random DNA level genetic changes going on, and selection works on that. They're both random variation and selection processes, but at different levels of organization. But another possibility related to some of the things that Mary Lynn was just talking about, but not necessarily involving structured water, but I want to talk about it in a more sort of generic way, that Darwinian way of thinking about things imagines that the information is naturally static and inert, and that it only changes by random variation. But if instead you think of the information as being dynamically maintained, so that a karyotype is not an inert structure, but it's something which is dynamically maintained by the cell, then when stress interrupts the processes of dynamic maintenance of that karyotype, it will cause many changes rapidly. But those many rapid changes won't be random. They will be specific and systematic to the kind of stress and the kind of interruption on those dynamic maintenance processes. So that rather than thinking of it as generating many random changes to karyotype, which then selection acts on, it's actually a systematic process of stress response, which is generating, yeah, sure, a variety, but not a random variety of responses to it. And then maybe that helps with the other problem of needing another individual with the same kind of changes in order for a species to be able to reproduce sexually, right? That if they were really generated at random, then the chances of that would be extraordinarily small. But if they were systematically creating a specific variety of changes, then the probability of that would be much more bearable. So I wondered, so it's interesting that you don't call it a random reorganization phase, you call it a chaotic phase, and a chaos, has sensitive dependence, but it's not random. So I wondered what your thoughts were on that distinction and whether there's any mileage there.
Yes, that's also a very good question and the thoughts. So they have two thinking of this one. So initially, we thinking because we actually did some calculation, like when drugs we treat, every cell is different, but what's the survival in the cancer? They even do not find a partner, right? So the simpler, they're just stable. So the stable is a much lower frequency, we know. So for example, if you, as we have so many flasks, how many can survive? We did all this calculation, and we found that it is much higher than the completely random. So it's much higher. But on the other hand, because 99.9% will be eliminated, and is not the non-randomness is also not, in terms of practically speaking, is also doesn't make sense as such. So we think they have two things. So like James Shapiro and many people, the thinking, it cannot be that random because it must be the involved, to certain stage. In that case, they are right is because we know certain gene cluster, they have boundaries easy to broken, right, than others. We actually try to do some experiment to calculate. But nevertheless, so the cell is conditioned in the way the active the pathway to disorganize the genome. That is for sure. But they are not sure which one can be survived. So you know what I mean? So therefore, so they have attention to destroy and create new, but they have no idea which one will be. So therefore, selection is still important in such a way to just help to select the winner, right? So, but you just say, I want to destroy the system. That is cognition decision, right? But who is winning, the out of control. But with the development, with the artificial selection, with all the stuff, the determinists become more and more, And then we actually can design. Right now we design we do the genome operation. We actually need to design certain configuration. So that's what happened initially. The Shanghai group, the fusion, the East chromosome, the 16 chromosome become 1 or 2 chromosome. The certain configuration is absolutely needed. You cannot just random put them together. They're not going to work. But certain composition, they are going to work. So that's actually that means that they do have the certain selection involved. But we not try to use typical neo-Darwinian to explain all of this, because they actually at the very beginning had a fundamentally mistake. So we do not worship that theory anymore. So we actually try to use a new way to.
Yeah, me neither, funnily enough.
Yeah, I know. So as such. So I hope I can answer your questions. So is they certainly have the biological involvement, but because limitation of the control of the environment, control the mechanistic, so they still have tons of randomness in it. So that's it. But with the future, with our information capability improve, and we actually can reduce such a randomness and get more. Yeah.
It seems like, if I'm putting a sharper point on what Richard said, it seems like your theory really, and I'm not sure how far still like you really walk down this trail, but it does seem like if there's system level inherited information at the karyotype level, and the karyotype is reorganized, then the program that reorganizes it has to live somewhere, right? And it can't be the result of a heritable program entirely, because it's reorganizing itself, and there can't be an instruction that says scramble me. And so that gets us, that sort of intrinsically or logically gets us past past the concept of an organism being strictly a heritable program of any type at any level, like karyotype plus genes or genes alone, but or even the whole cell as a heritable program. It seems like the more radical implication of your argument is that we have to throw out that entire thing, and we have to look for this other process that's a system level dynamical, process at the level of the self.
You can't have a Darwinian model. It's very well put, Charlie. You can't have a Darwinian model with an instruction of the form, which inherits an instruction of the form. Now I become a different me, right?
Do you think there might be a feedback loop between the somatic and the genetic, that is sort of like picking up when there's a crisis and the signal is coming from the somatic, it's time to reorganize genetically, or, sort of like the halves of the brain? Where's Ian McGilchrist when we need him?
Yeah, I mean, it certainly happens to brains, right? It certainly happens to brains that they reorganize in times of crisis. And so this is just pushing that down one level, I think.
Well, maybe more than one, but yeah.
More than one level. It's interesting that we can make these holonic arguments at multiple levels, isn't it?
Yeah.
Just in the context of this discussion, I was thinking, because I'm not aware if anybody tried to use to different formulation of entropy that could be maybe valuable in this framework. I'm talking about the Shannon entropy and the Demetrius evolutionary entropy. I don't know if you're familiar with this. I mean, this could be a way to using this concept, just a Shannon information theory of biological system can be seen as a DNA and entity that manage entropy and reduce, for example, the uncertainty through information encoding, like in the replication phase to maintain the fidelity, or they optimize efficiency through hierarchical organization, like it happened in the karyotype. So using the method of Demetrius evolutionary entropy that is rooted on the first law of thermodynamic, it will be maybe possible to give meaning to unpredictable pattern in the flow of information. Maybe this, I mean, the result that Harry is showing us could be modeled according to this mathematical solution.
So there are two difficult, and I just quickly thesis, and I will let Bill to ask a question. So we have difficult to use the Shannon's information theory because we actually thinking more. So the actual fundamental difference between the Shannon's theory, the physics, or the communication with biological, because for him, the idea is reduce the noise, right, to get to the but for biology right now we all about the noise, because without that we don't have all of the stuff. So our rational and the purpose is very different from Shannon's idea. So for example, if we accepted the gradual accumulation change, so that use the entropy gradually increase decrease is fine, but here biological system doesn't care how much waste of the energy is. They only care survive. So the survival and adaptation is totally different, concepts right now. We separate them as such. So in fact, like a human, right, is very adapted, successful, where the most waste of energy. We don't care the energy as such. So all of this concept previously we need to put it aside because it doesn't make sense anyway. So now we're actually thinking information is not talking about the minimum, the state, like the physics always, the stable always in the value, right? The evolution landscape, the stable survival always on the peak. So this totally different. What's the difference is because they did not consider information. So therefore, I don't think select entropy is a key in biology. In physics, it is, but in biology is not, because all the successful species is wasted energy. They do not, have very defined.
I mean, it's being used for the electroencephalographic analysis and is giving quite a good result, especially in a situation like PST disorder, major depression, attachment syndrome. So
sure.
There are quite a few of evidences that.
I give you an example. It's very difficult to calculate which one have entropy advantage, is a fox or rabbit. You just cannot calculate, right? So that's the thing. That's a biology thing. So that's why we think in biology we need a totally different physical theory. Even though I don't know what it looks like, but I know that's what we need anyway. So, but I will talk about later on to get some of the the tips.
Can
so
yeah. Oh, sorry.
Are
you done? Go ahead, Bill.
Yeah, thanks. Henry, thanks so much for coming. I'm a great fan of yours, and I think your work is brilliant, as you know, because we've talked together, or at least exchanged emails over the years. I just like to emphasize that everything that you've talked about fits really comfortably in cognition-based evolution. And I think discussing your work within that framework has an advantage because it places cancer into its proper context, which is it's a co-engineering process. So the basic premise of cognition-based evolution is quite simple. All cells are intelligent, which is actually undeniable, which means that cancer is intelligent. It means that it's an alternative self. All cells are a self. They have selfhood. They act to protect themselves. They act to protect their states of homeoetic preference. And all of biology is essentially a defense of that selfhood. Gene chaos is a creation of a new self, a cellular self. And that cellular self proceeds to problem solve and make decisions on its own in both collaborative and cooperative patterns with normative cells. Let's not forget that cancer is occurring inside us all the time. It's basically an everyday occurrence. Actually, it only very rarely gets out of control because there are mechanisms to suppress this new in our immune system. Occasionally, a new self occurs that is different, and it can co-engineer successfully. Let's not forget cancer is an ecological process. The tumor is an ecology. So is every metastasis. And so what you have then is, as you've elegantly shown, you get the first thing in cancer is a trigger that creates a new self that becomes a competitive entity within the larger self. And that trigger is generally stress related, but it doesn't have to be all the time. Because infectious disease dynamics means that every infection is a form of stress, and not every infection incurs overt physiological responses. Organisms and viruses go latent. We know what causes cancer, frankly. We know that viruses cause cancer. We've got every model. We've got HPV, we've got hepatitis C. We understand how viruses are one type of stressful trigger. So you can get some kind of a viral or subviral entity triggering a new self with or without any kind of environmental stress. And now you've got a new cellular self that is co-engineering in cooperation and, crucially, collaboration, codependency with normative cells. So the self is being dynamically maintained, as Richard points out so well. But what you do have is a non-random process. Both the normal cells and the cancer self are co-engineering through cognitive selection. They're choosing their tools, in which genes are tools. So then you have microevolutionary changes of the genes, which are adjusting to support the new self. And so I think your work is absolutely correct. I'll just finish by saying, you point out the most important thing about sexual reproduction, and it made me laugh because aside from me and my books and papers, no one has asserted, as far as I can recollect, Henry, I think you and I are the only two, that the point of sexual reproduction is to prevent substantial species drift. It's averaging. You cannot imagine, there is no simply better way to mathematically sustain an average than absolutely more or less dividing in half every single time over thousands of iterations. So sexual reproduction is obviously a means of attempting to maintain a species with the implied limit to vary over time as cognitive selection, as cells develop new solutions to new problems, form new partnerships. And so, Henry, I think your work is terrific, and it would obviously be a comfortable part of cognition-based evolution.
Thanks. Yep.
Can I restate the sexuality argument and see if I've got it clearly in my mind? So what the way I think of it is Shannon's theory of information is the theory of templates, right? So their information can be fit into, positional structures that are templated. And if we can do that with information, then we can compare one message to another and see if there are any mistakes, and that way we can correct errors, reduce noise, and that gives us, digital information, and that's correctable to infinity. And so it's a great way to transmit information faithfully. So as long as we're thinking of life as something that's organized by the genes, we can fit it into that Shannon, theory, and see how it's being error corrected, to preserve its integrity. But what you're saying is that at this higher level, the karyotype is also carrying information. And so it's only in the case of sexual organisms. I mean, they're multicellular asexual organisms too, but it's only in the case of sexual organisms that they're comparing one karyotype to another and correcting errors by eliminating, through sexuality, they eliminate deviations from the karyotype, right? And so I think the argument implies that there's a level of there's a level of sophistication of the inherited part of the instructions of life that's you need a greater fidelity, and that's why sexuality emerges in some places and not in others. Is that you need the more complex those inherited influences become, and the more complex the organism is, the more likely you are to encounter problems if you don't have some way of correcting errors in karyotype, because that's an important part of the system organization. And it also explains why it's more common in organisms that are more highly cognitive, that move around and change themselves more than others. So am I getting all that correct?
Are you asking? You're asking Henry, right?
Sorry, yeah, I'm asking Henry. Yeah.
Okay, good.
Yeah. Henry, can you hear me?
Yeah,
Oh.
I said okay.
Maybe I said too much. Maybe that was too much there.
So the key, what is the key part you try to clarify,
The key part is that Shannon's scheme is for error correction to fidelity, and that works great for genes, but it only works for karyotypes if you include sexuality, because that's when one karyotype is compared,
yeah, sure,
and error correct against the other one.
So, but then the situation is a little bit more complicated than you said, because the sexual, the correction of the order is the most powerful. But without those, right? So the asexual, the code maintained by physiological process, which is less powerful, but it's still maintained. Because if they actually change as a variable everything, so that fitness is changing, and therefore the selection plays the role to wipe you out, right? Or not. So because of that, in bacteria, they have so many horizontal gene transfer, right? But they're never going to be able to accumulate. So that's a key. So after 3.5 billion years, they come back and forth, they cannot. But as soon as you have the precisely maintain that one, and the biological process just go to complexity, just like that, right? So I wonder if this actually help you to articulate what you just said that one.
So you're saying that the innovation of sexuality led to complexity directly because it corrects for karyotype variation.
Yes, because it actually maintains the innovation. Because every time if you have innovation, by equilibrium, you just disappear, right? When you disappear forever because you need so many perfect stone. But when you have a perfect stone, when you have sex, then the perfect stone just become routine everyday life.
Right.
So that's how beautiful the system the system is, because it's a major form for the information preservation, makes all the miracle, very difficult chemical reaction, everything at once, and fix them become routine in biology. So biology is such a beautiful machine or system, not machine system, which can change this one. So therefore, from impossible to the routine, every time doing this kind of transition. So that's why they need the two phases of evolution. That's why the cognition, everything become so speed up the later stage of evolution compared with the initially. So the accumulation of information at a certain stage is a key for the new evolutionary, innovation.
Just to like steelman the counter argument though, you still do have a lot of complex organisms that are asexual and some that go back and forth, in different conditions between sexuality and asexuality.
But in fact, every time when they become asexual, most of them have sexual before, they're always on the tip of the evolutionary tree. So they're actually just right before be wiped out as such. Yeah.
Oh, what, asexuality evolves right before they're wiped out?
Yeah, many of this actually have the sexual and at the tip of the evolution, they all become asexual. So for example, the yeast, right? Same thing, yeast, so if you culture them and also, so they become polyploidy, they will be wiped out. But then if they become the meiosis, and then when they come out of this, so they still have perfect chromosome, so they can maintain as such.
Hmm.
So that's basically,
So
what happened.
So does this relate to parthenogenesis only happening in organisms that are in extremely stressed conditions and there are no sexual partners? Parthenogenesis.
I'm not sure how absolutely the conclusion is, but we know that every time like the yeast, right? So the stress always induces meiosis, and the stress always induces the cell fusion, right? So that actually is for sure. So we with the such, so so in the without, for example, some species, the whole summer is asexual, but right before they get into winter, they have sexual, so they actually preserve everything, right?
Okay.
So when the environment is, you can do everything, but when the environment is harsh, you have to preserve yourself rather than something else as such. So that could be, like the Bill mentioned, the maintenance of self. I mean,
interesting.
It's very important of information strategy because, but on another hand, now we actually, maybe Bill can get some advice about this one. So even though macroevolution is about the population size, everything, but when the crisis is coming, nature never care the carrier. They only care the information can go on, right? So that's the thing, no matter how dominant you are, you always replaced by new carriers. So that's another thing, it's we know how to put into the cognition, the story. Well, maybe the nature is selected smarter and smarter, right? As such,
Yeah.
So
that's really interesting. That they're new levels that carry information. Is that what you're saying?
yes, yeah.
Carry
Because so traditional Darwinian, they said, oh, there's no direction, right? But if there's no direction, why you digging the fossil, it becomes simpler and simpler, right? So that's the idea. So now we actually discuss with some physics, even Paul Davis or some. So we actually, like the idea, maybe the information become complex. That is a major driving force for all the evolutionary stuff, because overall evolution is built the complexity, right? So I think the cognition consciousness is just one of the example to showing that. So maybe that's the Bill's category of how to thinking about this, So we actually thinking, we imagine, so maybe the information is more basic than matter and energy. So that is explaining biology very well, because biology always uses energy to reorganize the matter, because different tissue is reorganize the matter, right? Different stuff. And different idea is reorganized above the matter, but still reorganize all the elements as such. So that actually, so if that's the case, we pay less attention of direct change from the information to energy to information. But through the biology, they actually beautifully change that, right? So we wonder whether if we can out of the space of the solar system, could we or should we change the dynamics of the equation of the universe as such? Yeah, yeah, please, Mary.
I'm wondering if anyone has been, probably not, because there aren't enough people studying the gene chaos and its implications, Henry, but I'm wondering if anyone is thinking of studying the patterns of the genetic chaos to see if there's fractality or any of the other sort of like structures that those who study the order within chaos are finding, and if there's what the patterns are in terms of gene chaos, because there's always some kind of pattern that can be observed, right?
Yes. So we don't know, we only know give enough time or enough space or material, you will be success. But we don't know what's this. For example, I'll give you an example. We actually did the one of simple question. So for drug resistant experiment, right? People always repeat, and so we actually had a hypothesis. We're thinking if you have the maximum killing power, you should induce the genome chaos. So if that's the case, the chemotherapy should be bad, right? So,
Yes.
so that's just a sort of experiment. So then we did the experiment. So we use cancer cell, use low treatment, this lower dosage treatment, higher dosage treatment.
Yep.
So clearly, if you use more higher dosage, you have survival cell is very less. So that when they come back, it always take a longer time.
Yeah.
But when we did the experiment, and the maximum killing actually come back the first, right? So this is a very, very surprise. And then we said, how could, if that's the case, why people did not report this phenomenon?
Right.
So then my student just repeat the experiment. Then we got stuck. We repeat once, twice, 3 times. Always we cannot repeat the come back quick, right?
Yeah.
So I asked my PhD student, we have to reach to the bottom. We repeat to see how many times they will come back. First time is 64 times they come back. Second time is 100 they come back. So now we know they are going to come back if you repeat enough time, right?
Yeah.
Then we did a second experiment because we use a small flask that did 100 times.
Yep.
Then we use a big flask, bam, 20 to 30 times, they always come back. So now we understand, because for the patient, they have billions of cells. So every time they come back. So now we actually understand this whole process. So we write the paper, like this 7 years now, never get published. People say that this is too much. If you
they've pushed the paradigm too far.
Yeah, because now we ask the question, how many times we need to repeat for biological experiment? This at least 100 times. So who did 100 times, right? So the people they still do not. But that is actually clinically true in a way. So therefore, the outlier rate, it is 1 in 100. But for all the biological science, we just wash away all the outliers because statistics tell us we do not pay attention.
Right.
Just this outlier kill the patient, right? So that's all that matters. So that's why all of this, the science did upside down right now. So biology, every time I'm thinking about all the key concepts have to be upside down, to.
Yes.
I have a question for you, Henry, about that, like the application of that cancer research. So I have read that fasting doubles the effectiveness of chemotherapy. And I don't know if you agree with that or if you know about that research or if you dispute it, but just proposing that that's true, I wonder if well, and then I mentioned another, like, just this is an N of one, just subjective experience that I've had because I've done a lot of fasting. And one year I fasted 65 days, like not all at once, but for during the year I fasted 65 days. And I experienced a lot of reorganization during that time, like mentally and physiologically. Um, and I feel like it helped me a lot. But I wonder if the combination of fasting and chemotherapy also stimulates more rapid cancer evolution. And so if it doubles the effectiveness, does it also shorten the time, that the cancer takes to recover and grow stronger? And I'm just an open question. I have no idea, but it might be something that would be interesting to look at because I think of that form of stress, as just it provokes another mysterious form of reorganization, um, physiologically.
So this is difficult question, right? We thought a lot. We actually apply to study. We never get a sense from NIH anyway. so that's the reality we have to face. But back to this, they have two difficulties. One is just we're talking about the cell, they have cognitive power to turn on some of different pathway, but they cannot predict the results. So for example, when the genome is unstable, right? So you use the stress that could kill more and more cancer cell. So many people should be okay, but some people unstable dramatically become sacrificed by such a treatment. So we just don't know who will have good response, who will have bad response, like by lottery, right? Some people buy lottery, they change their life for good. Some change their life for bad. Right? We consciously we don't want to buy that, right? But everything else is out of our control as such. So even they have such an effect. So we always thinking, like the chemo treatment, extremely good for very few, extremely bad for very few. And most people have generally bad, but they can tolerate bad results. So that's how we're thinking all the treatment as such. So therefore, in the future, we need to classify the patient. For example, someone's genome is very strong, we can push hard to kill the cancer cell. If some people is very fragile, right? We know mentally some fragile, don't push it. But for cancer, we just push it anyway. So it doesn't make sense as such. But so we need to classify people. In fact, we actually can do that to measure the blood, how random chromosome change is. But we cannot convince NIH people. Every time, always have one reviewer said, kill this proposal. It doesn't make sense. So
well, it stands out as an example in my mind because of the research that shows that fasting extends the lifespan of organisms. And I think of cancer as like this sort of fundamental process that shortens lifespans. And so if fasting is, tripling the life of a worm, and it's maybe for more complex organisms, it's increasing their lifespan, but not quite as much, then, how is that compatible with, just how does that interact?
So life is always complicated. It's difficult to get a conclusion. For example, I tell one people, one of the person I know very well, she had the higher hormone, woman's hormone, looking young at the age of 60, just looks like 40 or something. But then she got the ovarian cancer, and because of the high level of the hormone, right? That must be contribute to all that stuff. So you have no idea, which one the winning ticket you are getting, right? So for example, someone, looking younger and someone have certain price to pay as such. So especially knowing, even PP3 mutation have certain advantage, and you just, like the BRCA1, BRCA2, right, abrogenetically lethal. But if you at the same time knock out the PP3, they actually can have good life, even though the later stage they have some cancer. So this is completely, difficult. So we little bit hesitate to just, fully support or fully against some of the ideas, because it all depends, So that maybe is a new challenge for new science, because we cannot have such a certainty we had before. So that actually bothers people a lot,
Right.
So.
Totally. Great. Well, thank you so much, guys. It's coming towards the end of my time. I'm gonna have to go pick up my daughter from school soon. So, thank you guys so much. This was wonderful, Henry. This is fantastic, and I really
Your work is a huge contribution to the new synthesis, Henry.
yeah, and we
Thanks.
hope you'll
Thanks.
continue. We hope you'll continue to join us as well as we, keep going with this thread and hopefully try to learn from you and vice versa, as much as possible.
I'm hoping that many people will try to replicate your findings and start applying chaos theory and find what the fractal patterns are in the emergence of various cancers and types of cancers. I think that will make a really big difference in not only understanding life and the I think that what the fractal patterns will and the other aspects of this chaos will start to emerge in something that's more coherent and will give us more information about our agentic topics over time.
Thanks. Yeah.
Really
Thank you.
great.
Okay. Yeah.
Thanks so
Thank
much,
you.
guys. See you
Okay.
next month. All
Okay.
right. And thank you.
Thank you, everybody.
Bye-bye.
Bye.
Bye.
Then okay. That was really great, Henry.
Thanks. Yeah.
I have many
So-
more questions. I haven't I've read your articles and I'm reading the book slowly. I'm about halfway through, but I will have many more questions over time, I'm sure.
Okay. Okay. Great. Yeah. Just let me know, and we can
I will.
discuss.
Okay.
Okay.
Bye-bye.
Okay. Bye now.
Really, do you have a sec?
Yeah, have a sec.
Okay.
I think Steve's asleep over there on the couch.
Yep. Okay. So you can just
I'm gonna kill
close
the
out. You
recording.
can close out Stephen so that we can talk.
Let me stop.
Interrupting him.
Stop